Hi there, I'm Young, CTO of Monomer Bio. I've had an interest in active learning since my graduate days, and I'm excited to be able to present to this community. A month ago, Monomer Bio challenged a group of scientists, software engineers, and lab automators to grow a mystery cell culture on a robot. We believe this is the first event in which participants grew real cells on a robot in 24 hours. So I'm excited to present this to the symposium. The mystery cell line was announced at 10 a.m. on the day of the hackathon, and we spent the morning setting up teams and giving high-level tutorials. By noon, we had participants writing code and growing cells. And overnight, cells were fed, passaged, and measured overnight by the robot, and then 4 p.m. were presentations. We put up an event page and we hoped for the best. We honestly didn't know if anyone was going to show up. As you can see from the photo here, there's a lot of interest. Over 130 people signed up, and this included an ML engineer and lab automation engineer from AI companies, like Lila, that flew in all the way from Boston to San Francisco. We also had lab automation engineers and scientists from traditional biopharma, such as In Sitro, Big Hat, Icon, Merck, Genentech, a tech bio investor who's also flying in just for the demo day. And due to the space constraints of the Monomer San Francisco office, we could only let 6 groups of 5 people participate. And none of them had any experience with our robotic work cell or our software. And remember, none of these folks knew the cell type until the actual day. So what was the mystery cell culture? It was Vibrio nitrogens. We chose this cell line for 2 reasons. One, for practical hackathon considerations. It's the fastest growing cell line known to man, and we wanted to get as many growth curves in 24 hours as possible. We used an engineered strain from Novo Bio. This is a strain that's already been optimized, and it's a really fast growing cell. It's also a very scientifically interesting cell. Because of its fast cycle time, there's an increasing amount of interest in using it as a model organism, as well as a chassis for growing and producing other products. It's just not as well studied as E. coli, and Novo Bio, which provided the cells in the media, were actually very interested in the results and seeing how they could further optimize and engineer this chassis. So how did we grow the cells? So Monomer, we provided robotics to grow the cells. So this is a robotic work cell on the right, and the robotic work cell comprises of a robotic arm, as a KX2 arm, a plate reader to measure absorbances and to monitor cell growth, a liquid handler to change media and to passage the cells, a heating incubator to keep the cells at the desired temperature of 37 degrees Celsius, and cooling incubator to store the media, as well as a carousel to hold consumables like plates as well as tip racks. And these are all off-the-shelf instruments. In Monomer, we develop software, and we don't develop specific instruments. And our software is designed to provide a fast, intuitive UI to monitor cell cultures. So this is our UI, and this is what made it easy for all the participants to monitor cell concentration as robots are growing cells overnight. Each square here denotes a well, and different colors here denote cell concentration as measured by OD600 through a plate reader. You can hover a well and see its growth curve. You can also see how metadata, like how many times it has been passaged, as well as trace its lineage to its parent cell culture, so on and so forth. For the execution purposes, we decided that MCP interface would be best for the hackathon. This is going to be used by the first time by different first-time users, and they only had 24 hours, so we decided that a more simplified chat-based interface would make sense. So this is our MCP interface, right? And it's on top of our existing API that allowed participants to use natural language to query data from the cloud, use natural language to command the on-premise work cell, and also a couple of the participants used the MCP server to develop AI agents. Since this was our very first hackathon, we opened up a very limited set of capabilities compared to what our API and the robotic work cell is really capable of. So the MCP specification here is more of a distillation of the key functions with 2 main interfaces. One's the cloud interface, which interacts with our cloud-based culture monitor to list cultures, plates, and get culture details. We also had the on-premise automation platform, which is labeled work cell control, that allowed users to use the natural language to list the culture plates that are available, create their own workflows, as well as to instantiate those workflows. For the next hackathon, we'll expand on this set of capabilities. But already with this limited interface, we learned that cells plus AI plus robot tech plus an excited group of nerds equals a lot of fun. From this video, you can see that we have a robotic work cell moving plates around and executing experiments. We have a UI to watch progress and see the cell lines racing each other. So from this UI, the different participants in the hackathon can look at the different cultures, pick the best one to passage, as well as choose and mark them as ready to passage via the culture status. Once they've been set, they can then filter for the ready to passage cultures. And from this list, they can then export all the cultures that are ready for passage via a bulk CSV export. They can then paste this to an MCP interface to control the work cell, and here They tell which the scientists use the MCP interface to tell the work cell which wells to passage, how many wells, and in what dilution. So for the first day, the UI was used to kickstart some initial growth curves and let participants get used to the interface and robotics. Then AI agents were developed to keep growing the cells overnight, 100% autonomously, as you can see here. So here's actual footage of the work cell growing cells overnight in a complete cycle. Results were presented back on Sunday, and interestingly, participants also posted about the projects on social media since the hackathon. And there's a lot of interest here, as you can see. A couple of highlights I'll link here. There's a LinkedIn post. There was a post by a principal software engineer at Genentech, an automation engineer from Lila AI, a head of R&D, a research associate, even the CEO of Potato AI, which is one of our partner AI companies, described he felt something special happened in the hackathon. Dive deeper here, we represent the results from two teams, a team called Vector AI and a team called Skill Me Maybe. So Vector AI, they separated the workflow into distinct phases with multiple agents throughout the process. There was an explore, constrain, and recommend phase, which were fully implemented and allowed them to fully generate initial step suggestions from user prompts. For the execute, monitor, analyze, and recommend loop, they only had time to implement it with still a human in the loop, driving the next set of experiments while chatting with various agents. For this audience, in particular, for the active learning audience, the recommend phase here was an entirely LLM-driven, in this case Claude-based interface. This is not a traditional active learning process. As you can tell, the methods are completely opaque and non-deterministic, but they still got some results from these iterations. And here you can see that they have multiple agents that the user can talk to, and the hypothesis was that a multi-agent system with agents specializing in experiment design and literature research can provide a distinct advantage over using a single agent. And here you can see how they're querying their literature agent for questions about Vibrio. And next here you can see how they're querying our workcell MCP server and asking what they can do in the workcell, and they're learning from MCP server what possible experiments they can run. So you can see that they can look at the different workflows and different methods that's available to the MCP interface. Unfortunately, they only managed a few iterations, so nothing conclusive was drawn from this approach, but I thought it was interesting to see how far we can push LLM-centric approaches at the moment. The next team was Skill Me Maybe. They used one of the AI partners, Potato AI, to research existing protocols and to come up with an experimental design for the workcell. Given the focus of the symposium, I'll highlight that this team used Bayesian optimization. I don't have access to the specifics such as the prior acquisition functions or the sampling strategy, but I'm sure they'd be delighted to talk to you about it. However, they identified key liquid handling parameters, such as mix cycles, volume, and height, and by the end of the hackathon, they had a human-in-the-loop system, which was continuously learning from each results of each experiment. And here's the video from Skill Me Maybe, the presentation that they did. So you can see here how they've managed to get optimal parameters based on the results that they had generated using the workcell. And through some correlation analysis, they've identified the key factors as well as the best parameters, the best values for these parameters for this iteration. And they can then suggest new optimized parameters for the next set of experimentation. So as you can see, it's really exciting to have these two teams participate in a hackathon, and they had adopted active learning approaches which would be relevant to this audience. So a couple of next steps for us. So December through February, we intend to run more close-up experiments with select collaborators. We'll provide the dojo, you bring the AI or active learning optimization algorithms. The goal here is to publish more in-depth case studies, not just get it working in 24 hours. And in March, we plan to host Hackathon Round 2. So the last event was done in collaboration with 3 other AI companies, briefly Cypher and Potato, and the Master of Ceremonies was Michael from the Bay Area Lab Automators. We're looking for collaborators that are closer fit to our vertical focus in cell culture. For example, on our AI, and we'll open up a wider set of capabilities to our workcell. If folks from the active learning community are interested, either to participate in growing cells themselves or offer their optimization tools to participants, please connect and let us know. Too many folks to acknowledge in this talk, but I'll put this slide up for you to read. I would have loved to attend this live, but unfortunately unable to do so due to existing personal commitments. Please send me an email and I'll be happy to respond to questions as best as I can. Thank you.